Thank you for the introduction and for the invitation. And nice that you saw the paper about the Scorpius. Today I will only focus on the Pseudoscorpions and present the my progress on the PhD thesis with the title Identification of Bioactive Compounds from Pseudoscorpion Venom. And this project I'm doing at the working group of Professor Predel at the University of Cologne. And so at first I will start with a short introduction about Pseudoscorpions, as these animals are not really well known in the public. So for two reasons. So first, they are living in crevice habitats, very hidden from us, and they are really small. So the species I work with, the house Pseudoscorpion, has a body length of around 5 millimeters. So don't get distracted by this image. These animals are really tiny, and as you can see from the external habitus, they look like a scorpion without the stinger. And interestingly, within Pseudoscorpions, another unique venom delivery system has evolved, as these animals use their pedipalps to simultaneously grasp the prey and inject venom with venom glands that are located inside the shielded fingers and the shielded hand. And as you can see, the Pseudoscorpions, they are capable of paralyzing prey, which is similar in size as the individual Pseudoscorpion, as this fruit fly. And then the liquefied prey content is sucked out with the chelicera, as it is done in case of scorpions and spiders. And for me, the exciting part about Pseudoscorpions was that when I started working on them, really nothing was known about the venom composition. Not a single venom compound was identified. And this is what I wanted to change with this PhD project. And the first challenge was to obtain the venom in a sufficient quantity. And for this purpose, we have designed this extraction device, which can be used to immobilize the animals under this layer of foam plastic and simultaneously apply electricity with these movable arms at the integument of the shielded hand. And in the beginning was quite frustrating for me because the electricity alone was not sufficient to trigger the release of venom, but it was necessary to move one of the shielded fingers into a liquid-filled capillary. And only this, in combination with electricity, causes the release of venom, as you can see here. So this is then a really tiny drop of venom, but finally it was enough to perform an analysis of the venom composition. So for this purpose, we performed a comprehensive analysis using proteomic and transcriptomic data. And finally, we could identify 124 potential venom compounds, which are here classified into cysteine-containing and cysteine-poor. And we further classified these based on similarity to compounds from the online database UniProt. And in the end, we could identify all major substance classes which can be expected in a predatory venom. So we have this fraction of potential neurotoxins, and these are now the first neurotoxins identified for Pseudoscorpions, which have a confirmation by proteomic data. And among these, there are, for example, some compounds with similarity to potassium channel toxins, and we also found several enzymes. And these are mostly phospholipases and metalloproteases, which are very commonly found in many predatory venoms. We also found antimicrobial peptides, which I will talk about later on. And we have this huge fraction of novel compounds, which did not have a significant hit in the database. And this we also expected to some degree, as there are no Pseudoscorpion venom compounds in the databases, and venom is a fast evolving trait, as we heard before. So yeah, then we knew, or we had a first impression of the compounds that are present in the venom of the host Pseudoscorpion, and in the next step we wanted to know what are the effects caused by the venom. And for this purpose, we used oocytes extracted from xenopus, expressed with some specific ion channels, and we applied the crude venom to these oocytes and measured the effect with the 2 microelectrode patch clamp method. And yeah, these are the results. So in this first step, we only tested on a few selected ion channels, so not a really full range of ion channels. And we could find an effect on the Shaker IR channel from Drosophila. So this is a potassium channel, and here in the presence of the venom, the ion flow through this channel is reduced. So the venom causes an inhibition of this channel. We also tested against the sodium channel from the varroamide, and in this case, the venom causes a modulation of the inactivation process, as the ion current through this channel is sustained. And this was especially nice for us because the house Pseudoscorpion is used as a pest control against the varroamide. And here we could show that the venom has indeed an effect on one of the channels of this mite. So in the next step, we also wanted to test the activity of some of the antimicrobial peptides of the house Pseudoscorpion's venom. And so for this purpose, we obtained some synthesized peptides. So actually, these antimicrobial peptides, these were the first compounds we identified in the venom of the host Pseudoscorpion, and they were classified as antimicrobial peptides based on a similarity to AMPs from scorpion venom. And we wanted to see if these peptides indeed have an antimicrobial activity. We also tested for insecticidal effects by using aphid feeding assay, and we also wanted to know if there are cytotoxic effects to mammalian cells. So here are the results. So for this checkered scene 1, so in this study we have tested mostly checkered scene 1, and these other peptides here, they are fragments of this major checkered scene 1. And in the aphid feeding assay, we could see that checkered scene 1 has a strong effect on the survival rate of aphids, which is comparable to the effect caused by a commercial insecticide, the spinosad, which is here in gray. So the checkered scene 1 has a quite strong insecticidal effect. And yeah, so here you can see the effect on the tested bacteria, and also in this case the checkered scene 1 was really very efficient in killing bacteria. So these are the minimum inhibitory concentrations, and so we tested against several indicator strains which included gram-negative bacteria like Escherichia coli, Pseudomonas, but also gram-positive strains like this multi-resistant Staphylococcus aureus. And in all cases, this peptide was quite efficient in killing these bacteria. And these values are even comparable to the minimal inhibitory concentrations caused by gentamicin, which is a really highly efficient antibiotic. So this was a quite nice outcome, but to evaluate the potential for drug development, as I mentioned, we also tested the cytotoxic effects. And so here we have an intact cell culture of mammalian cells. In this case, these are kidney cells from a dog. And if you apply checkered scene 1 in the highest concentration we tested to the cell culture, this has a quite drastic effect, as it causes a nearly complete disintegration of the cell culture. And these so the concentration or the concentrations causing these cytotoxic effects, they were at least higher than, so 1 magnitude higher than the lowest concentration causing the antimicrobial effects. But yeah, due to the cytotoxic effects, the potential for drug development is limited. Okay, so with that, I come to my summary. So far, we were able to identify the major fraction of venom compounds for the house Pseudoscorpion. We performed first activity tests on cellular level, and we found these strong antimicrobial, insecticidal, and cytotoxic effects for the first synthesized Pseudoscorpion AMPs. And with that, I want to thank you for your attention, and I'm happy to answer any questions.
Yeah, thank you very much, Jonas. Any questions from the audience? Else I would start. Do you have the sequences also of the full toxin enzymes you investigate? So via transcriptomic data or just the MS data?
So yes, I have the transcriptomic data. So I have the full sequences of also of the enzymes, so also of the metalloproteases. And so at least for those I showed here. So I mean, I didn't show the sequences, but I also have so these were all confirmed by proteomic data and by transcriptomic data. And
do you saw any similarities to known toxins, or are they kind of like totally different?
So there were similarities to known toxins. So because that was more or less the basis for classifying them as a metalloprotease, for example. I don't really remember from which species the metalloproteases were, or the other enzymes, because I didn't focus too much on the enzymes, but at least they had similarity to known metalloproteases and phospholipases. So yeah.
Okay. And we have a ton of questions in the chat from Farrington for you. So why do you use oocytes? The yolk tend to absorb toxins, so they are not as nice as CHO or hex cells.
So I mean, I was working in cooperation with the TÜTGAT lab in Leuden, and they have this toxin tests quite established. So I didn't perform these tests on my own, but yeah, I think they are quite advanced with the oocytes, and yeah, so actually the other methods, yeah, might have also been a good idea, but in this case, yeah, the cooperation partner I was working with, he was relying on the oocytes still. Yeah.
What other ion channels will you take into account?
So in this first tests, we tested mainly, I think it was only these 2 ion channels we tested. So these tests were, as I said, done by a cooperation partner. I don't know if he also tested additional ion channels which didn't show an effect, but in the first step, we only tested the venom on very few ion channels. The reason was we expected there should be an effect on insects. So for this reason, we chose the shaker IR channel, and we also expected an effect against the varroa mite as the house Pseudoscorpion is used as a pest control. So this was only a preliminary testing, so we didn't include much more channels.
And do you plan to do structural analysis or go into silico?
So far, this is not planned yet. I mean, maybe in a future project, but at the moment, I'm focusing on fractionating the Pseudoscorpion venom. So that's at the moment my focus. because we want to confirm these effects we measured on the oocytes also for single isolated compounds. And that's at the moment my focus. And afterwards, of course, I would also do further structural elucidation, maybe, but right now this is not our plan for the next period. Yeah.
Cedric also offered you, so if you are interested into testing them on calcium channels, you can just contact him.
Ah, that's perfect.
And Leah's amazed that you got these tiny guys milked. What is the amount you get from one?
So actually, it's not that easy to estimate. So we said it's around 5 nanoliters, because those capillaries I was showing, those were pulled capillaries, so they had a really tiny tip, and to quantify the exact amount, is quite hard. So at least I always have 1 microliter of liquid inside the capillary. So to give you a relation, and then there is this really tiny droplet of venom going into this 1 microliter. So I estimate it's to be 5 to 10 nanoliters. So a really tiny amount.
Is there anything known about the structure of the venom apparatus? So just one of the pins that has the outlet, right?
So there is not that much known. We performed some electron micrographs, electroscanning micrographs. So from the external, we know something at least. So these pseudoscorpions, they have a structure. They have a hair, a setae, close to the venom tooth, which is called lamina defensor, and we think that this structure, or this was also hypothesized in the literature before, is responsible for this effect that we don't get the venom by electricity alone. So it seems, at least to me, that it is decisive that this lamina defensor is in contact with liquid, so that these pseudoscorpions can ensure that the prey is successfully penetrated. So that because when I, for example, moved the finger only into a capillary, there was no liquid inside. There was also no venom coming out. Only when there was liquid inside was working. So and for me, this was, of course, in the beginning quite annoying, because it took me some time to figure this out. Because for spiders and scorpions, it's just you turn on the electricity and the venom is coming out. And for the pseudoscorpions, it was not like this. So this was, I would say, some weeks of confusion, and then at some points, I had the right idea to add the liquid. So.
But that's a super interesting mechanism, so that you always need the liquid that it's get anyway injected. Yeah, so this is really, I have the hypothesis is because the pseudoscorpions, they do 4Z, so they attach to beetles or something to get to another place. And if they would always lose venom, if they pinch into the beetle or something, this would be a problem, because it's also a matter of energy cost. And so my idea is that they have this mechanism to prevent that they lose the venom by the time they attach to some beetle or to anything. Yeah. But this is, at least so far, it's not really investigated. So how this lamina defensor, this setae, is working. So there's no publication of this.